make note of what I am telling you is going to be on your lab practicum. And if it were I would get on the computer, find the picture, put it on my laptop, and be very familiar with it, because you will see it as in slides on your lab practicum. However, Karen doesn't put that stuff out until the very last, the last class that we have. So you don't have much time to study for it, but I'm going to tell you as we go by. Okay, so all fungi are chemoheterotrophs. That means that they're living on preformed organic matter. And they excrete extracellular enzymes, and they make the enzymes inside their cell, export them outside of the cell, because let's say you have a fungi living on an orange or a piece of bread in your house. They can't get the bread in to eat, it's too big. So they send out enzymes to start breaking down these big macromolecules into smaller components that can come across the cell wall and that they can use for their nutrients. So they're major decomposers of dead plant material. And in that way then they're going to help put back into the, I don't know what word it is, put back into the earth these broken down particles to be used again by other organisms. So when you're talking about enzymes, most times they will end in an ASE, and also most times, but not always, that word describes, you know, what it's breaking down. So, for example, the fungi makes an enzyme called cellulase, and that enzyme is the one that will break down cellulose. And cellulose, or you know, you find that in plants and your wood, and they break it down to glucose that they can use for energy. Glucose is a simple molecule. They can bring it across their cell wall and use it for energy. So most molds are aerobic. What does it mean when they're aerobic? They got it. Yeah, they're using oxygen. Or when we're talking about yeast, they are facultative. Most of them are facultative anaerobes. What does that mean? They can, if they want oxygen, that's their favorite. But if it isn't there, they're able to metabolize things or ferment things without oxygen. So this is a little kind of hit the anatomy of fungi. So the body of a fungi is called a thallus, and it consists of long filaments of cells that are joined together called hyphae. I think I've drawn these for you. So you can have a septate hyphae. That means it has a cross wall, actually cells. However, those cross walls have little pores in them, so nutrients can go back and forth in those cells. And they usually have 1 to 2 nuclei per cell. So that is a septate hyphae. Then they have one that doesn't have any cross walls. It just has a bunch of nuclei in it, and that one is called acinocytic hyphae. Or your hyphae. And these hyphae grow by elongating at the tips. And they also can fragment, and then a fragment can start a new hyphal growing too. And there was a scientist up in Oregon that did research on this fungus up there, and he measured this one hyphae, and it was like, I can't remember if it was, he did squares of an acre, but it was just one hyphae that took up all this area. You can look in your book, I think it's in there. So now we have 2 types of hyphae. One is called the vegetative hyphae, And that's the one that obtains the nutrients. And then we also have aerial hyphae. So off this vegetative hyphae, the fungi might grow up a stalk and this is an aerial hyphae. And a bunch of these hyphae that would grow together is called a mycelium. So look at your mushroom stalk, that's a mycelium. That's a lot of single-celled hyphae in it. Yeast are unicellular. When they reproduce, they can reproduce by fission, and that's where a yeast cell will divide evenly into two cells, or they can divide by budding, and that's where the yeast forms a bud on its surface, and that bud's going to elongate. So yeast are kind of like a little bit oval like that, so and make a bud. This bud is eventually going to be another yeast cell. So the nucleic acids in the yeast are going to divide, and they're going to go into that bud, and the cross wall is going to form. You know, one replication will go in the bud, and the other one will stay in the original parent cell, and then that bud's going to break off and form a new yeast cell. These cells, they're unicellular, but once in a while, they make what's called pseudohyphae, and that's where these buds, instead of detaching, they make like a kind of like, it's not a real hyphae, but they make kind of like a hyphae. And one yeast that does this is Candida albicans, and it attaches to epithelial cells as a yeast, but if it wants to invade your tissues, then it forms a pseudohyphae so that it can get down into those tissues. And you don't have to copy this, just telling you. Yeast are capable of facultative anaerobic growth. They can do aerobic respiration. That is where the yeast and oxygen are going to, in the presence of oxygen, are going to metabolize carbohydrates to carbon dioxide and water. So that's what aerobic respiration. If they do anaerobic respiration, the yeast doesn't have the oxygen there, so it's going to ferment the carbohydrates to ethanol, which is an alcohol, and carbon dioxide. So we use that. Anybody that's made bread knows you have to get brewer's yeast to put in the bread so that the bread will rise. We're using this anaerobic respiration that these yeasts do to help us with the food and wine and beer industries. So here's a picture of Saccharomyces cerevisiae. What is it? What would you use that one in? Deer. And those little pink things that are on there, they've colorized this, but the little stumps there, those are bud scars. That's where a bud has been, and then when it breaks off, it leaves a scar on the yeast cell. Dimorphic fungi, most of those are pathogenic. Dimorphism, di means two, so that means that it has two forms that it can grow in. It can either grow as a yeast or as a mold. So it will grow as a mold if it's at room temperature. 25 degrees Celsius is about room temperature. But if it will grow as a yeast if it's at 37 degrees Celsius. What else is around 37 degrees Celsius? Body part, yeah, your body. Life cycle, filamentous fungi reproduce asexually or by fragmentation or spores. And they reproduce sexually by spores. Fungal spores aren't as hardy as endospores, but they can survive hot, dry environments. So let's talk about the asexual spores. They're formed from that aerial hyphae that we drew there. They're produced by one fungus when we're doing an asexual spore, just one organism involved. And so when that spore germinates, it will be identical to the parent cell. And a type of asexual spores are conidiospores. You can have uni or multiple spores, and they are not enclosed in a sac. So I drew one of them for you there. And they are produced in a chain at the end of the conidiophore. So now this, what we're saying this whole structure is an aerial hyphae, but this part is the stalk is conidiophore, and the spores are conidiospores. And when we change to something else, it'll still be something spore and then something spore. So an example of fungi that make these conidiospores are Aspergillus, and that can be a pathogen, and penicillium. Here's a picture of Aspergillus, and you can see those spores. This is a conidiophore, the stalk that the spores are coming off of. And here's those individual spores. Here's a picture of Aspergillus in media. What does that mean? You start your computer.
Push postpone.
What? Huh?
Postpone.
Change the time first.
How
Let me do it.
Change the time first.
Wait. There.
4 hours.
Oh, what is going on? I don't like. See, I'm hooked up to the school's thing. Otherwise, this wouldn't be doing it. Again, some more showing you the spores. Ow. And again, you can see the nice little head there that they're coming off of, and more conidiospores. Also, in your book, you should look at these because they have pictures there, and then you'll understand. There's also what's called arthroconidia. They are formed by fragmentation of a septate hyphae into a single thicker cell. So an example of a fungi that does that is Coccidioides immitis. That one is definitely a pathogen. This is the one that my sister came down with, is Coccidioides immitis. And it causes pulmonary mycosis. Blastoconidia is formed from buds coming off the parent cell in some yeast cells. So examples of that would be Candida albicans and Cryptococcus. And Cryptococcus is a fungi that also can cause pulmonary mycosis.
Pulmonary mycosis.
Yeah, fungal disease. Mycosis is a fungal disease. Mycology is the study of the fungi.